very
Great.
much for coming.
Thank you, Jordy. So first of all, I would like to thank Jordy for hosting me here for my sabbatical. I was really excited to go away on a sabbatical and be somewhere different for a while and meet some new researchers and learn some new things. So I'm really happy to be here for that reason. Today I'm going to talk about some research I've been doing in the past few years, which mostly concerns hydraulics and water relations of plants. In my career, I've worked on photosynthesis, I've worked on nutrients, but we've had several big droughts, both in California where I work and also in Panama where I have a joint appointment in the past few years. So my research program has really moved in the direction of drought and water relations in the past 3 or 4 years. So my research basically falls into 2 disciplines in ecology. The first is plant physiological ecology, which deals with individual-based measurements on individual plants, looking at their environmental responses, often in an evolutionary context, and often what we're measuring are fluxes of matter and energy. And I relate this to ecosystem science because in ecosystem science, we're also looking at fluxes of matter and energy, although over larger scales very often. And this often concerns biogeochemical cycles. And then at very large scales, this determines where plants live in terms of plant life zones, which brings us back to physiological ecology. So my research has always kind of been kind of interdisciplinary between these 2 areas. I'm going to talk today about several of the different sites that I work in. And so I put up pictures here so you can see some of my sites. This is Amazonian forest in French Guiana. It's the newest site I've been working in. I've only been working there for about 4 years, so it's kind of the most exciting. It's like a new toy. It's a really fascinating site. It's eastern Amazonian forest, so they're very low nutrient soils. The forest is about 30 meters in height, and it's a really well preserved forest site with a lot of infrastructure at the station there. This is the site in California that's closest to where I live. It's about an hour away from where I live. It's a desert chaparral ecosystem in Southern California, and it's right on the border of where chaparral grades into Mojave Desert. So it's really on the dry side of what we consider Mediterranean ecosystems, and this is what it looks like. There's a really big variety of different growth forms in this forest. This is a tropical dry forest in Mexico at Zibilchaltún in the Yucatan Peninsula. And I did a postdoc there many years ago, and I've continued collaborating there on and off. It's kind of the only tropical dry forest site that I work in, and it's a really fascinating site. I've really enjoyed my work there and continue to work with some researchers there. And then this is Barro Colorado Island in Panama. This is the site where I did my PhD work, and I've continued working there. I have a joint appointment at the Smithsonian there. I spend time there every year, and so it's kind of the site of all of these that I've been working at the longest, that I kind of consider my home base. And so I'll be showing you a little bit of data from all of these different sites today, because these are basically the places where I work. So I'm talking about drought, but what is the context of what I'm talking about? And I kind of try to put this into a bigger context to understand why this is important. And so if we think about this, atmospheric CO2 is increasing, and this has been linked to extreme climatic events such as drought. And when drought happens, sometimes there is mortality of species in terrestrial ecosystems, but who dies and who lives depends on species drought survival traits. And that's what I'm going to be talking about today. And this is important in a large context because if we have mortality of trees in an ecosystem, this also causes a biomass loss, and as that biomass decomposes, it releases CO2 through decomposition back to the atmosphere. So all of this is kind of linked in a global manner, and I kind of view my research in this little box here, kind of contributing to the whole thing. So I'm going to talk about drought survival traits among species, and the question for today's talk is really what seems to be a very simple one, is how do trees survive drought? Okay, and a few years ago, my students and I, in talking about this and thinking about what different species do, we figured out that people looking at different traits actually had different answers of how plant species survive drought. So we decided to start making a list of all of the traits that we consider to be mechanisms for how plant species survive drought. And every time I give this talk, someone comes to me after and says, you know, I have this trait that's not on your list, or I think one of these traits shouldn't be on your list, or something like that. So if that's the case, come talk to me afterwards and we can have a coffee, or you can buy me a glass of wine and we can talk it out. Okay. So how do trees survive drought? Well, I have this list here. The first one, and probably the most well studied, is having a cavitation resistant xylem. So in the stem where water is moved, some species can actually withstand very low water potentials, very dry conditions, and not lose their ability to move water. Whereas other species, when things get a little dry, their xylem vessels stop functioning. And so there's a lot of variation in species. This has been studied a lot, probably the most. Capacitance is another one, though. And capacitance refers to how plants move stored water around their hydraulic system. And so if one part of their hydraulic system is suffering, how they move water to alleviate that stress is characterized by capacitance. Okay? Drought deciduousness is another one. You know, when it gets really dry, plants lose a lot of water from their leaves. So if things get really dry, some plants will just lose their leaves and cut off that loss of water. That's a fairly effective method for surviving drought. It's also been shown that during drought, some plants close their stomates so tightly that they stop photosynthesizing and they actually die of carbon starvation, even though water is the limiting factor. And so we've been studying photosynthetic stems in the desert for the past few years, and we've kind of suggested that photosynthetic stems may be a way that plants can survive drought, because if they lose their leaves, they can still gain some carbon through their green stems and maybe not die of carbon starvation during a drought. Okay, one of the most obvious is deep roots. As things dry down, plants with deep roots and access to water can often maintain physiological function during drought, whereas plants with very shallow roots may get dried out more quickly. So this is a really basic one. Regulation of gas exchange is an important one and somewhat hard to characterize. Plants open and close their stomata during the day, and when there's a drought, they can regulate how much they open it and how much they close it. And some of them have fairly complex regulation of when they open their stomata and when they don't. So we've been theorizing that plants that open their stomata to gain carbon when conditions are right. Maybe in the morning when it's cooler and they lose less water for the amount of carbon that they gain, would be more effective during drought at still being able to do some photosynthesis, but not losing as much water when they do that. And so really maximizing that balance is important. Okay, effective osmotic control. Plant cells can control their osmotic concentrations. They move ions in and out of the xylem actively, okay, and their ionic potential of their cytosol helps them withstand low water potentials. So to the extent that plants can do this effectively, they can actually probably deal with drier conditions. And then finally, when the drought is really strong, plants have no water source, they've lost all their leaves, the epidermis of the plant, the skin of the plant, actually loses water at very slow rates. Okay, even if all the stomata are closed, at very slow rates they lose water, and so that rate varies among different plant species. And so when plants have no water and they're losing water slowly, it really just depends how fast they lose water before they may completely desiccate. And so this is probably one of the least studied traits on this list, but there is some data accumulating on this now. Okay, and we've written about this in a couple of recent papers, and the point here is that what I call a drought survival strategy is really the composition of these traits. And so, you know, a plant may have good regulation of gas exchange and deep roots, but not a cavitation resistant xylem, or some other combination. And so depending how plants combine these different traits really determines what I call their strategy. Okay. Excuse me. Okay, so very quick review of water transport in trees. I know everyone remembers their plant physiology class very well, but just to kind of orient everyone here. So plants take up water in their roots, and water moves passively along a water potential gradient up to the leaves and then out into the atmosphere. So the atmosphere is very dry relative to plant tissue, and so the water basically just gets sucked all the way through the plant out their stomates. No metabolic energy is expended, okay, and water moves through a variety of tissues in the roots and the leaves, which have lots of living cells, but in the stem, they're mostly moving through vessels, which can be xylem vessels or tracheids, but they're basically tubes, okay? And so really within the stem, it's the physics of that water movement that controls when the xylem fails during drought and when it functions well. And so that's really the big difference, where we have a lot of living cells here and here, but really just kind of dead cells that are tubes. It's like a bundle of pipes. Okay, and that's the main plumbing of the middle part of the stem, and that's often where things go bad. Okay, let's take a little closer look at the xylem. And so these are some diagrams of tracheids and what a vessel member looks like. They're basically tubes. They have these little holes on the side that are called pits, and those allow water to move in between vessels. Okay, and if we take a look at our classic xylem vessel here, you can see there's kind of this tube. There's a plate at the end with little holes, okay, but it's basically an empty tube. And there are these little cells on the side called companion cells, and those are alive. Okay, and those companion cells, they're living cells, but they're a very small proportion of the actual xylem. Most of that xylem is actually these empty tubes. So it's really the physics of what's happening in these empty tubes, not so much the biology within the stem that controls the xylem function. Now, when things get dry, bad things can happen. Okay, and this is an example of that. What happens when things get dry in the xylem? And so in this example, this is one of these pits. It's one of these little holes that connect xylem vessels, these little holes on the xylem walls. And what they have is kind of this opening, and there's this little membrane here. And what's been shown is that if there's a big pressure difference between the air and a water-filled vessel, you can get air sucked through, and you can get embolism, basically a bubble forming, that will block that xylem vessel. It'll cavitate that vessel. And this often happens because the water in the xylem is under tension. So it's under a negative pressure. And the way I usually think about this is, if you have a very thick milkshake, a very thick batido, and you suck with your straw and you don't get anything, and the straw starts to kind of collapse. Okay, that's kind of what negative pressure is. And so with the xylem being under negative pressure, air can get sucked in and block that vessel. And then water can't move anymore. Okay, and so that's a problem. And different plants have a different water potential at which they will fail. Okay, so every species has kind of a different level of tension that they can stand before air gets sucked into their xylem and then it can't function anymore. And so us as ecophysiologists, this is what we like to measure, because we can say, aha, this plant species will fail at -2 megapascals Some example of data I've collected, these are 14 canopied tree species from the Amazon and Parakou, French Guiana. This is just published this year. And you can see that for these species, there's a good amount of variation in this. These are all dominant canopy species in the Amazon in a site where there's about 200 species per hectare. Okay, we measured the 14 most common ones to try to characterize what was going on for as much of the forest as we could, which is actually a relatively small amount. And this is what we found. There's a good amount of variation. There are some species that are extremely vulnerable, like Licania here, and there's some species that are much more resistant, like Epidua falcata here, and there's a lot of variation in between. And because we staggered, we stratified our sampling by wood density, so we measured the lowest wood density and the highest wood density in the plot, and then a variety in between. We think we captured a lot of the variation at the site. And so that was kind of our best way of capturing a lot of variation when we're only measuring 14 out of 200 species, okay, at this very diverse site. So it does tell us something, but there's clearly a lot more to learn. This is that same data in a bar graph, so that you can see the values. Species up here are more resistant. They don't lose half their conductivity until higher tensions. Species down here are more vulnerable. So this is Licania alba, the most vulnerable of the species, and then Epidua falcata, the most resistant. Okay, and then there's a variety in between. A lot of them kind of fall in the same space here. This data has been hard to measure in tropical forests because the trees are tall, it's hot, working under difficult conditions, but we've managed to do this for a good number of species now. And how do we do that? Well, first of all, we have Jocelyn Casal, who climbs trees. And he will climb to the top of a 30-meter tree and cut down a 2 to 3 meter branch for us that falls to the ground for us to measure. And he will do this on 4 or 5 trees before noon. He is amazing. Okay? And nowhere have I worked with someone like him who can climb trees and get samples. It's really amazing. It's the only way we'd be able to do this work in the Amazon. Okay, and so he was great to work with. And then once we get our branch to the ground, we bag it up so that it stops losing water, and we bring it back to the lab. And this is about a 2 meter or 3 meter tall branch in this bag, about the size of a body. And this is where our research starts to look like an episode of Breaking Bad. And I don't know if I was more terrified that it looked like a body bag, or that the police looked that we had 5 of them in the car and just let us go. So, I'm not sure what I would have done if he pulled us over trying to explain that we had trees in these bags in French, would have been kind of difficult for me, but they didn't do anything. So we take them back to the lab, and then we sample them little by little as they dry down to generate our vulnerability curve. Okay. Once we get back to the lab, things look kind of like this. We have stems that are hooked up to a system of tubing, and we're pushing water through there. We're measuring water flow just with a pipette and a stopwatch, so fairly basic measurements. And we were able to measure these 14 species in this way in this lab in French Guiana. So that's basically what it looked like there. In Panama, things are different. We have a canopy crane. I'm not sure if you can see this, but there's actually a construction crane in the middle of the forest here. And so this is the executive way to get samples. You barely walk in the mud, and you go up in the canopy crane, and with the walkie-talkie you tell the operator where to take you, and you collect your samples directly from the upper canopy, hanging from the gondola. Okay, this was January of 2017. We did some work there. This is my student Aleida Acosta, and it was very easy. With maybe 6 trips to the crane in a month, we were able to measure the same amount of species that we did in French Guiana over 4 months. And so much faster, much easier work. And Panama has 2 of these, and we've sampled both of them. So just an example of some of that data. These vulnerability curve data from Panama. Here we measured 6 trees and 6 lianas. This was led by my former graduate student, Mark de Guzman. And we were able to show that on average, the lianas are more vulnerable than the trees. The trees here are in black. Lianas tend to lose their ability to conduct water at tensions that aren't very negative, whereas the trees are more resistant. Now there are a number of trade-offs. It's thought that lianas have deeper roots, and so it's okay that they're vulnerable. They may have better access to water. The data on that is spotty, but we are definitely able to show that they're coexisting in the same forest, and the lianas are more vulnerable than the trees. So, excuse me. For our site in California, in our desert chaparral site, the interesting thing here is that there's a lot more variation. So we have species such as juniper that are really resistant to cavitation. Some junipers are the most resistant in the world. And then we also have species like Rusovata. This is it's a tropical affiliate. It's an anacardiaceae that grows in Southern California. It's extremely vulnerable, but they have deep roots and they tap the water table. So within the system, we kind of have a little bit of everything, and we have a much wider span than we do at our wetter tropical sites. And so that's kind of something, as I compare different sites, I see a variation in the breadth of these traits, which is interesting to me. Okay, now I mentioned capacitance, okay, as another way that plants can survive drought. And capacitance really allows plants to kind of survive shorter term droughts. So during the day, if it gets hot and there's a low tension in part of the xylem, a plant with high capacitance can mobilize its xylem water to that area to alleviate some of that tension. And it's been shown that some plant species have a high capacitance and they don't have a very resistant xylem. So to some degree, high capacitance can compensate for a vulnerable xylem. And this is measured. It's the slope of this line at the initial part of the pressure volume curve. And so the pressure volume curve is basically generated by taking a little piece of wood and alternatingly measuring its water potential and its weight so that we can calculate the water released versus the water potential. And it's very much like if you do a moisture release curve with soil. Okay, so it's very analogous to that, but it's with sapwood instead of soil. And this initial slope before it loses turgor basically gives us our capacitance. It's the amount of water that can be moved per unit of gradient in water potential. Okay, and so it gives us an idea of that as things dry down, how effectively water can be moved around to different parts of the sapwood that have different water potentials at that time. And with these curves, we put them through an elaborate spreadsheet that allows us to calculate a number of traits. It allows us to calculate the turgor loss point. And so if you remember, there are these companion cells that are living in the xylem. So they're living cells, so they have a turgor loss point. So we're able to characterize their turgor loss point, their osmotic potential at full turgor. We get the capacitance out of this and several other traits that allow us to characterize aspects of the sapwood and the xylem that are not captured by our vulnerability curve. So it's really a different analysis, and it takes into account some of the role of the living cells and their ability to osmotically regulate parts of the xylem, whereas the vulnerability curve really gives us the physics of what's going on. These measurements are much more biological, and with both of them we have a lot more knowledge. My former student Mark was able to show that species with greater capacitance have greater water status at midday. So this is from one of the canopy cranes, and this is our combination of trees and lianas. The lianas are in black here. Okay, and so capacitance on a day-to-day basis is helping compensate for water potential drops at midday. Okay. And an interesting thing to me about capacitance, this is data that I just analyzed relatively recently. Excuse me. You know, measuring all of these physiological traits in xylem for drought resistance is difficult. Takes a lot of time. And so what a lot of modelers do is they just use wood density as kind of a proxy for this, with species with dense wood being more resistant and species with light wood being more vulnerable. And in some sites, this works really well. I've seen some very good relationships with this. However, in our sites, it doesn't seem to work very well, because if we look at P50, this is for the Amazon site, and this is for our site in California here. There's really not a significant relationship between wood density and P50. So if you were to use P50, if you were to use wood density to try to predict P50 in these sites, you wouldn't get very far. But for capacitance, the relationships are actually quite good. Okay, so we have significant relationships with capacitance. Now, much less is known about the relationship between capacitance and mortality. So while it can be predicted by wood density, we're still kind of exploring what capacitance tells us about a species' drought resistance. But I thought it was interesting to point out, if you're gonna model based on wood density, you're gonna get much closer with capacitance than P50 very often. Okay, drought deciduousness. Many species in the tropics lose their leaves during the dry season. Okay, they do it every year. Whether it's raining or not, at that time of year, they lose their leaves, just like trees in the temperate zone lose their leaves when it's cold. Okay. However, there are also species that even during the wet season, if there's a drought, they will start to lose their leaves, and they will eventually go leafless, even if it's the wet season, if there's a drought. Okay. And so that's different from the annual programmed deciduousness. It's the ability to shed leaves as a way to reduce losses of water. Okay. And there are also species that during a drought will hold on to every single leaf until they turn brown, and they will die with their leaves on. Okay. Why they do that, I don't know. We clearly see all. Tropical forests are very diverse, so you can see all the different ranges of strategies. And so deciduousness is something that we've started exploring. How prevalent is it? How dynamic is it in different species? How flexible is it? And especially in tropical dry forest species, we've seen there's a lot of opportunistic behavior. So a little bit of rain in the dry season, some of them will grow back some leaves. And things start to dry out in the wet season, they may lose a little bit of leaves. And so for some species, there's actually a good amount of variation and some ability to change it, whereas for some species it's very programmed. It really depends. We followed this for a year at our desert chaparral site, and we found all kinds of different behaviors. Here, the black bars are when species have leaves. The white bars are when they don't have leaves. And the gray bars are when they don't have leaves, but they have photosynthetic stems. Okay, and so you can see every species pretty much has a different pattern here. And there's only 2 months or so where everything has leaves. For the rest of the year, things are varying a lot in terms of when they have leaves and when they don't. And I think part of it is in the site, we have species from the tropics that tend more towards the dry season deciduousness. And we also have species from the temperate zone that, even though they're in a desert, they're still kind of programmed to lose leaves when it's cold. And then we have evergreens. So we kind of have a little bit of everything in this site. And so we thought that this was going to tell us something, that they were going to be separated into very clear classes. And they're not. It's almost like every species is kind of doing its own thing. But the fact that there are so many different strategies tells me that this may be something that plants are using to regulate their water loss during different parts of the year. Okay, photosynthetic stems. My former graduate student, Elena Avila Lovera, worked on this for her thesis from the perspective that photosynthetic stems could be an effective way for plants to deal with drought. Once they lose their leaves, they can still gain some carbon without, their leaves losing a bunch of water. And so she studied this in the deserts of Southern California, and came up with a lot of really good insights about what's going on there, comparing different species that had photosynthetic stems with ones that didn't. And one really interesting analysis that we did was looking at our site in the Amazon and using databases to look out of the hundreds of species there, which ones had photosynthetic stems and which ones didn't. And whether this might be somehow advantageous in tropical forests during short-term droughts. And the interesting thing we found was that in this Amazonian forest, only 3% of the species, 3.5% of the species had photosynthetic stems. So not very much. However, when we took a closer look, we found that these 3.5% of the species with the photosynthetic stems were the most abundant species. And so when we looked by basal area, we found that about a third of the species actually had photosynthetic stems. And this is, for some of them, it's in the main stem. They have streaks of photosynthetic tissue, but a lot of them, it's in the newer growth. So branches down to maybe, I don't know, 6 or 7 centimeters in diameter actually still had green skin on them. And so, so they tended to be much more of the common species, which made us think this could even be an advantage in very wet ecosystems like tropical forests for helping gain carbon if they start to shed leaves during a drought. And so this is something we're looking into further, but this was kind of interesting preliminary analysis that we did looking at this. Okay. What's next? Deep roots. Rooting depth in plants is very hard to measure. It's one of the hardest things because we can't see them, and any way that we do it is usually kind of destructive. And so one approach we've used is using stable isotopes of hydrogen or oxygen to try to estimate the depth of water uptake. Okay, and what we do, so this is a soil profile here, and what we see is that as things get deeper, the oxygen isotopic composition of the soil water actually becomes more depleted in the heavy isotope. So right near the surface, there's a lot more of the heavy isotope because there's more evaporation, and the lighter isotopes tend to evaporate more easily. And so we get a fractionation in the top of the soil. So it's heavier in the top and it's lighter in the bottom. And this gray bar is that's well water. So we know that that's what the groundwater is because we can get that from a well. But we basically have this profile. So we can take a little bit of xylem from the plant and extract the water and look at the isotopic composition and kind of estimate what the depth of water uptake is for that plant at that time, because this is something that can change. And so it's always interesting what you find with this. Sometimes the results aren't very clear, but we've been able to learn things that I think we could not learn with any other method. For example, we learned that trees and evergreen trees had differences in their rooting depth early in forest succession, but that late in forest succession, there was no difference. So early in succession, the evergreen trees put down much deeper roots, and the deciduous trees have much more shallow roots. And this kind of makes sense. If the evergreen trees are going to be photosynthesizing throughout the dry season, they better have some deep roots to be able to get water. The deciduous species, they lose their leaves in the dry season, so they're not investing in very deep roots. Okay, but that goes away with forest succession. So late successional forest, there's no difference. Okay, at that point, they've all had a chance to grow deep roots. Also, we learned that among just the deciduous plants, there was a difference in rooting depth based on size. So bigger individuals had deeper roots, smaller individuals had more shallow roots. Okay, with evergreen trees, there was no difference at all in any of the forest stages. And so, although these techniques are difficult, they did help us learn some things that I really don't think there's any other way we could have learned. So it kind of gives us a different class of information about the access that plants have for water and kind of where they stand in terms of what they might do during a drought. Now, we also tried this several times in our site in California, and this site is much more difficult. It was hard. You can see there's this blip in the soil here. We've seen this several times. We don't know when it is, and I think it's because the rainfall there is very pulsed. So there'll be like a heavy rain, and then it won't rain for a long time. And then there'll be a heavy rain, and then it won't rain for a long time. And so you get these kind of pulses of water moving through the soil. And then also, the colder the storm is, the more depleted it is. So if we get more tropical moisture coming through, the rainfall is going to be more over here. If we get more of a northern storm coming through, the rainfall is going to be more over here. So in addition to the evaporative surface, we also have a lot of different water sources. So it's harder here. And because of this terrible blip here, we weren't able to get that much information. But we were able to basically classify species into two main groups. So things that are very shallow, things that are very deep, and then some of them kind of in the middle that we couldn't tell. But still, it gave us a lot of information. We did this with our 15 species, and this gray bar is basically the range of where species were. So anything that kind of fell in this range here, we were not able to kind of characterize, because it could be here, it could be here, so tricky. But we've continued to do this, so we have more samples to analyze. Hopefully we'll get some good, clearer profiles to help differentiate what's going on with some of these species. Okay, I also mentioned regulation of gas exchange. So this has been done at our site in Amazonia for a number of species, and this was done by single rope climbing technique. So actually like climbing up to the tree with a pyrometer and measuring gas exchange in the wet season and the dry season. And so for most of the species measured, it's exactly what we'd expect, right? This is stomatal conductance, and you would expect stomatal conductance, more transpiration from the leaves, more open stomata in the wet season, and then maybe kind of closing down a little bit in the dry season, okay? So yeah, most of the species we measured did that. But there were some species that just kind of had low stomatal conductance, whether it's the wet season or the dry season, they don't care. And then we had some species that actually increased their stomatal conductance from the wet season to the dry season. And we thought, well, why would a species do that? The only thing we can think of is that during the wet season, it's very cloudy. And so there's not that much light. During the dry season, it's more often sunny. And so these species maybe are taking advantage of more sunny conditions during the dry season and opening their stomates more to gain carbon because there's more light energy. But then only a few of them were doing it. So again, lots of different things. It's a very diverse forest. And in many of these studies, we kind of imagine that there's going to be clear trends, and we find that different species are doing very different things. Okay. My former graduate student, Alex Pivovarov, also measured regulation of gas exchange throughout the year in California. And this was during the Great California Drought. So between 2012 and 2016, we got the least amount of rainfall in a 4-year period in the history of California. It was extremely dry for those years. And one year, at least in Southern California, we completely missed a wet season. It didn't rain all through the dry season. It didn't rain all through the wet season, and then another dry season. And so through that time, we had a couple of mists with fog events, but no real ground soaking rains for about a whole year and a half where I live. So this was extremely dry conditions. There was a lot of mortality. And Alex followed the stomatal responses for 1 year of species at a chaparral site. So these were chaparral shrubs. And excuse me, what you can see here, there's data for 17 species, and then this graph is everything together. And so, so what she found, she was really looking for the point at which stomata close. What is the water potential at which stomata closes among these different species? And she found that point, it varies with the hydraulic safety margin of the plant species. It varies with their P50, which I was showing you before. And so there's kind of a coordinated hydraulic system of the stem and the stomata in coordination. The more resistant the plant is, the more it can go before it closes its stomata. So that was one thing that she found. She also found that there were some species that weren't really closing their stomata until below -8 megapascals. And we published this year. This was the lowest ever shown point of stomatal closure. Okay, no other species has been shown to keep their stomata open until water potentials that are that low. And so really remarkable under these conditions. It's both the fact that these species are very drought resistant, and we were measuring them during the biggest drought that they've ever seen. And so really unique data set on regulation of gas exchange. And this is a very rich data set. We just published this, but I think there's a lot more within this data set that could actually be published once we go through all the species and all these curves and their behavior throughout the year and the seasonality, and there's really a lot there. So, second to last trait, effective osmotic control. Okay, and this one I think doesn't get as much attention among ecophysiologists as it should, because it's really a cellular process. Plants highly regulate, their cells highly regulate ionic control. They keep a bunch of ions in their vacuole, and for each ion, there's particular carriers that move those ions between the vacuole and the cytosol, and they're all under kind of coordinated regulation in a cell. So it's really kind of complex what they do. These are measurements of the water potential at turgor loss point and the osmotic potential at full turgor. So these are from my xylem pressure volume curves. They're derived from that. Both traits are related to wood density, and basically what we see is that species that have a lower osmotic potential at full turgor, so their osmoticum is allowing those cells to hold a more negative water potential, they also have a turgor loss point. So they can hold on to turgor until more and more stressful conditions. And so this is kind of direct evidence for this benefit of osmotic control. Plants that have more ions in their cytosol basically are going to withstand lower water potentials before they lose turgor. Okay, but I emphasize that this is a dynamic trait. So we measured it once, but plants have a lot of regulation about this. So I wouldn't say that measuring it once really captures the full story. It's evidence, but there's a lot more going on here as well. And finally, cuticle conductance. So there's a drought, the plant loses its leaves, closes all of its stomates, and there's this slow rate of water loss from its epidermis, from its skin. The plant skin loses water slowly. And so we investigated this in the stems of a number of species in the desert in Southern California. And what we found was really interesting. So especially among the plants with photosynthetic stems. And so this is mass-based photosynthesis of green stems. Okay, and this is their dry season G-min. It's their minimum conductance. Okay, so it's basically cuticle conductance. We do it in low light, so the stomata close. We put them under a fan, and we just measure how much water they lose very slowly. And what really, the reason I show this graph is for two reasons. One, it shows you there's a relatively big range in the cuticle conductance. And so just to emphasize, not all species loses water at the same rate through their skin. Okay, there's a lot of different characteristics about their epidermis that determine how much water they lose passively, but also that it scales with photosynthesis. So if we think of stem photosynthesis as a benefit, here's the cost. Stems that can photosynthesize more, even when they close their stomata, are going to lose more water. Okay. So, so stem photosynthesis may be a benefit in the desert when there's a drought, but there also may be a cost to it. So again, weighing that carbon gain versus water loss during drought is always really a key thing for figuring out how plants are going to survive drought. So, conclusions and future directions. My biggest problem is that there's so many species with so many different strategies, and these measurements take a long time. And so it's very difficult to kind of get enough data to get a whole picture of what all these species are doing. And it's very important to do this because it's the only way to add realism to the models that we're using to predict when plants are going to die of drought. It's a complex problem that we still cannot predict very well what type of drought is going to lead to mortality events, and if there's a mortality event, which plants are going to die, which plants are going to live. Okay, so I advocate any new approaches that can get us more data. Stable isotopes gives us new types of data. Spectral reflectance from satellites can give us new types of data that are different, well, similar to physiological data, but at different scales. Molecular approaches like transcriptomics can give us new kinds of data. And so I really advocate all different kinds of approaches to get more data on this problem. And also increasing the throughput of physiological measures. any ways that we can increase the speed of our techniques to be able to measure more species and get more data and get a more comprehensive picture of what all these different species are doing would help. And for me, maybe this is a bias from having worked in tropical forests, but the biodiversity is always the biggest challenge. There's just too many species to measure. Thank you very much.
Thank you very much. Luke, questions? Yeah. Well,
thank you. You always talk about, you always have 1 point for one species, but what's the intraspecific variability? Does it exist? Do you think it's insignificant?
I think it's actually very important. When we look at one site, it varies a lot. For some species, there's more variability than others, but usually the variability between species is greater than the variability within species at one site. That changes when we start looking across sites. And so that's where I think it becomes especially important. But I think that within one site it also deserves more attention, because even within one species, if there's a big drought, some individuals of that species may live and some may die. And whether that's related to their intraspecific variation in some of these drought resistance traits, it probably is. And so, so I think it's very worthwhile. And I think there are several projects I've heard people talking about. I think Jordy's working on this now, of trying to characterize that, because for, as long as I've been doing ecophysiology, trying to just characterize a species has been the same thing, but people are asking that exact question now. So I think there's going to be a lot more data coming on that soon.
Yeah, please. So I have a question for you in order you can enlighten our research. When we have drought deciduous plants in Sub-Sahel, for example, we see that they accumulate water just before the raining season. Is there any mechanism that come to your mind to explain that? Is it a kind of your capacitance, but not daily, but seasonal?
I would guess that they're deep rooted, and that they have some storage tissues, like the bombaceaes. They have a storage tissue where they can actually increase their water storage before a drought, and they also have photosynthetic stems, that kind of soft tissue on the outer bark. And so plants that either have deep roots and storage tissues can do that, and I would expect that they're probably doing it with osmotic control. So they may lower the osmotic potential of those cells in the storage tissues and increase the water coming in before the dry season. I know that, at least I'm familiar with the kind of ceiba and bombaceous trees of Latin America. I guess in Africa, like baobab trees may do the same kind of thing.
Well, they do that before the raining season.
Before the rainy season.
That's really curious, yeah. They seem to know. And then they have the leaf unfolding ready when the rain starts.
Oh, so they can get a jump on the wet season carbon gain. Yeah. They must have deep roots.
Yeah. Your explanation seems logical, yeah.
I would think so, yeah. Yeah. That's curious.
That's curious. That's seen by your reflectance measurement that you are operating for.
Uh-huh. Yeah.
The other thing is about lianas. You know that there is a hot debate on that. So you tell us that lianas, or I don't know this right spelling in English, lianas you say, or lianas?
Lianas, yeah.
Lianas, they are more vulnerable than trees.
Mm-hmm.
And still in your hydraulic or your physiological variables, parameters, they seem to tell us the contrary.
Mm-hmm.
And because they are increasing continuously in these environments, despite decreasing drought, so there is something that doesn't seem to fit. So
yeah, the story goes, according to my colleague Stefan Schnitzer, that because they depend on trees for mechanical support, that saves them resources that they can allocate to growing deeper roots. And his big evidence for this is if you look at dry season growth. So if you look at dry season growth between lianas and trees in the same site, the lianas grow a lot more during the dry season. And so it suggests that they have access to water, and they're using that to gain carbon when it's not cloudy and when sun is available, and they grow more in the dry season, and that must mean they have deeper roots. If you look at the literature, everything's been found. It's been found that they have the same rooting depth. It's been found that they have deeper roots. It's been found they have shallower roots. And so I think there's not enough data, because it's really been looked at in only a few sites that I know of. And so, yeah.
Thank you very much.
Thank you.
Thanks for the talk. You have shown us like several traits. If you had to choose like one or two, because if we want to add like more realism to models, which ones do you think that better capture the drought responses?
Wow. Well, the first one, I talked a lot about P50 in the beginning.
Then you forget about it.
Yeah, right. I think that's a really important one. It's hard to measure, but it really tells you when the stem of the plant is going to fail, and that pretty much equals death. So I would say that one's really important. And then for a second one, I would say rooting depth. You know, rooting depth really, you can have a vulnerable xylem if you have really deep roots. And so between those two tells you a lot. So those would be my two.
And have you tried to see the relation between some of these traits with growth data or mortality data?
Yes. So for the French Guiana data set, I'm working with Bruno Hérault. He has the long-term growth data at that site. They've been measuring diameters once a year since 1981. And so he has all that data. It's a lot of data, but he has my physiological data, and we're looking to see if there's any correspondence there. Other groups have done this in the 50-hectare plot in Panama, and I've been associated with some of that work. But there isn't as much physiological data. It's being accumulated now, but it hasn't been done there yet. And so when that comes, and a number of other plots, people are kind of doing the same thing, but haven't completed it yet. So I expect it soon.
I have a question and a comment.
Yes.
The question is, you have a lot of information on different physiological traits for different species. Have you related this with mortalities of these species? Because you have explained how the different physiological variables are related, but eventually you are talking about mortality. So my question is, have you related this with mortality?
That is the key question. So in Southern California, during the Great California Drought, none of my species showed elevated mortality. We thought we would see something because we already have the hydraulics traits measured, and in 4 years, none of those species showed increased mortality. So we weren't able to say anything except that they're really hardy species.
So I understand that your motto was survival, and your presentation, the title was more related with survival.
Survival, yes, that's right. Also in French Guiana, we're looking at that now because we have long-term data there. In Panama, we're looking at this as well. We collected a set of data for, I think, 15 or so species this summer, and we specifically chose them as one set that showed elevated mortality during a very large El Nino, and the other set that didn't show any elevated mortality. So we chose them specifically for their mortality classes and measured their hydraulic traits. So my graduate student is hopefully analyzing that data right now, and we should have an answer soon, hopefully.
Okay, and then my comment is, especially in the first slides, when you relate the mortality trees, the tree mortality, abundance and duration, with a more general context, I think that we need to incorporate also not only these physiological traits, but demographical. For instance, in the list of the mechanisms for survival, why not resprouting? And resprouting is in the middle. It's between the demographical and also some kind of anatomical and eventually physiological response. But also the life history, because you may have a high mortality, that if you have a high recruitment later, okay, there is a carbon loss by the biomass loss, but there is also perhaps new CO2 income. So I think that we need to go further and try to figure out a kind of syndrome incorporating both these physiological states, but also this demographic of life history, or as any word you want to use.
That's a great comment. Resprouting is one I had not thought about. So that's one that could potentially make it to the list after further deliberation. But then also your point about different life stages. So all of my measurements here pertain to mature individuals.
Yeah.
And so when you look at seedlings, it's kind of a different story. It may be even a different set of traits. And so...
No, I understand,
yeah,
but if we want to scale up to... This is the reason I'm referring to the first slide, the general context. When we want to scale up to the ecosystem or the community level, we need to incorporate also this demographical process, not only to focus in the physiological one. I agree that your work is great, and I like very much all the insights, all the overview. But this is just this is the general.
Yeah, no, you're right, and if you have some time, I'd like to talk about how to do that. I'm not a demographer, but I could think of the different life stages and how something like this might be incorporated, or we can explore it. Yeah.
Okay.
You mentioned before the two of your favorites being P50 and P40. Now, my question is always, what's the cost of improving the trait to improve the resistance to drought? So with deep rooting, it's clear it costs carbon to build the tissue to root deep. But what about P50? What's the cost?
The cost, actually, there's a pretty well-known trade-off. Vessels that are wider tend to cavitate more easily. So when you build a vessel that's very resistant to cavitation, it's usually more narrow, so the rate of water flow is lower through there. And so that's really the cost. Otherwise, all species would have very resistant vessels. But if a species wants to grow fast, it has to move water to its leaves very quickly. It has to have bigger vessels, and those are going to be more vulnerable. So that's really the cost, which actually ties into some life history strategies with fast versus slow growing species. Not perfectly, but there is a cost. So.
Well, thanks for the great talk. I really enjoyed it. It's nice for me to see that you advocate for using more stable isotopes. I couldn't agree more because I've been using isotopes, carbon and oxygen, and I think it's very powerful approach to somehow, like using them as also proxy for reconstructing physiological traits. And my question is whether you looked at the delta fertility and try, because you have all these hydraulic traits, and it would be nice to look also at the water use efficiency and whether you may relate some of the difference in water use efficiency to the traits that you mentioned.
Yeah, we have in all these studies looked at leaf and for photosynthetic stems, also the photosynthetic stem delta carbon 13. Those were some of the most interesting data. So we found good correspondence of the stem versus leaf photosynthetic tissue related to their water use efficiency. For the California work with just the leaves of the shrubs, we found some extremes, but in the middle it didn't tell us much. And then the tropical forest work, it really didn't tell us much at all. There was so much variation, and I think in the tropics also the source CO2 gets mixed with the soil respiration, and so those numbers were all over the place. I could never get any pattern out of that. So we tried to varying degrees. We've also tried with the leaves the oxygen isotopic composition and found some patterns, again, mostly with the studies in the desert with the leaf versus stem photosynthesis. And so it kind of seems like the drier the ecosystem, the more information that we get, because there's more of a span of behaviors. And so, but it tells us something. Yeah.
Okay, I actually had a question, but I also have a comment related to Benny's, well, Benny left already, but Benny's question. Actually, it seems to me that something we don't consider enough maybe is that also embolism, so we see it in a negative way, and of course there are good reasons for that, but it's also a way that for the leaves to sense the conditions under which the plant stem is. And it would be very difficult for many species to close stomata without losing any conductivity on the xylem, because the water potential will never get low enough probably at the leaf level. So I think that there is also a trade-off, or something that explains this variability in P50s has to do with this. And actually my question, which is also related to Paco's and Tere's previous questions, is, so I like your list of traits, even more when you add the sprouting capacity, but I think that I think it's very nice, but the key to me is really how do we combine them, because it's true that just having one of them can really make the difference, but in many occasions it's not enough. So it's really the combination, and I think we're starting to understand a little bit better how they combine and the trade-offs involved in the relations between the different traits. But I think, to me, really that's, if we want to improve the models, that's where we need to go, to really understand how these different sets of traits, how the syndromes that combine the traits somehow arrange and describe the strategies. So I don't know if you have ideas on where to...
Yeah, so in our California work, we were expecting, like, 3 or 4 classes of strategies where there's kind of these coordinated sets of traits that always occur together, and we found that wasn't true. The only ones that really tend to combine are species deep rooting and P50. So if something is deep rooted, it can have a vulnerable P50. If something is shallow rooted, it must have a resistant P50. But everything else, like photosynthetic stems, well, the deepest rooted species and the most shallow rooted species had photosynthetic stems. And for drought deciduousness, it was all over the place. And so I don't know if it's just that site or what, but I haven't found really structured classes using my whole list of traits. There's only a couple that tend to occur together. And so I think we need more data, because there's actually very few sites that have characterized all these traits. And so we've done it in California. We're trying to do it in French Guiana. I think we can, pulling data from different studies. But it's a lot of different things to measure. And so, just need more data.
Okay, thanks.
Thank you.
I forgot to mention something. Well, as always, we will be having dinner with you. In this case, actually, it will be tomorrow. Not normally we do it the same day, but on this occasion, it will be tomorrow, Thursday, and also with Patrick. So anyone that wants to...